So, what happens when you smash together the code of life with the power of modern computing? You get bioinformatics. And believe me, it's one of the most exciting fields in science today. We're going to dive into how we're learning to read and maybe even write the very instruction manual for life itself. To really get what's going on, we have to rewind a bit. Think about this. The very first time we sequenced a human genome, the Human Genome Project, it cost a mind-blowing $3 billion. That's about $1 for every single letter in our DNA. An incredible achievement, for sure, but at that price, it wasn't exactly something you could do every day. Okay, now fast forward to today. That $3 billion price tag, it's plummeted to less than $1,000. That is a 3 million-fold drop in cost. It's just wild. And that technological leap has completely opened the floodgates, creating this massive tsunami of biological data. The problem then became, what do we do with all of it? Well, to make sense of it all, we had to start thinking about biology a little differently. Not just as wet lab science with test tubes, but as digital code. It's literally like a programming language, but instead of running on a computer, it runs you. And that's exactly where bioinformatics comes in. It's the toolbox, all the software, the algorithms, the stats that lets us read, search, and actually understand this gigantic biological library. It's the critical bridge that takes us from mountains of raw data to actual meaningful insights about life. Now, if biology is a language, there's a basic grammar to it, and it's called the central dogma. It's pretty simple, really. Think of your DNA as the master blueprint locked away safely in your cells. When your body needs to do something, it makes a copy of one part of the blueprint. That's RNA, the message. And that message is then used to build a protein, which is the actual machine that goes and does the job. Blueprint, message, machine. That's the core flow of life. So we've got a blueprint, and we're starting to understand the grammar. But how in the world do we actually read the thing? This is where the tech gets really, really cool. This really shows the whole revolution in a nutshell. The original method, Sanger sequencing, was kind of like a medieval monk carefully transcribing a book letter by letter. It was super accurate, but unbelievably slow and expensive. Next-generation sequencing, or NGS, is a completely different game. It's more like taking 1 million copies of the book, putting them all through a shredder, and then using a supercomputer to piece it all back together. Sounds chaotic, right? But it is incredibly fast and cheap. But that amazing speed created a new kind of challenge. I mean, we're talking about a book with over 3 billion letters in it. How on earth do you find the one specific gene, or maybe even one misspelled word that you're looking for? It's a classic big data problem. The answer was a tool that absolutely changed everything. BLAST. You can think of it as the Google for biology. You can literally copy and paste a DNA sequence, and BLAST will scour global databases to find anything that looks like it. And it's not just one tool, it's a whole toolkit. You can compare DNA to DNA with BLAST-N, or you can use BLAST-X to see what kind of protein your DNA might build. It's an absolutely essential tool for making sense of it all. And this isn't just some academic exercise. Being able to read and search our genetic code has profound real-world consequences, especially in medicine. We're now shifting from just reading the code to using it to develop actual treatments and cures. You see, a lot of diseases are caused by what are essentially typos in our genetic code. A bioinformatician's job is to be the ultimate proofreader. They're scanning for things like a single nucleotide polymorphism, or a SNP, which is like a single spelling mistake. An indel is like a word that's been added or deleted. And a copy number variant, that's when whole paragraphs get copied or deleted by mistake. Finding these typos can be the key to diagnosing a disease. But, it's not just about the static code. We can also measure how active genes are. Using a technique called RNA-seq, we can see if a gene is turned on or off. Take a look at this chart. In the healthy sample, the activity for gene X is super low, but in the disease sample, it's cranked way up. For a researcher, that's a massive clue, pointing them right to what might be going wrong. Okay, so we've covered reading the code and finding the typos. Now, let's look at what's next, the future. And that future is being written as we speak, powered by artificial intelligence. Just look at how the pace of discovery has been accelerating on this timeline. From figuring out DNA's double helix to mapping the entire human genome, it's been an incredible ride. And then, in 2020, something happened that was a total leap forward, a true fusion of AI and biology. That leap was AlphaFold. Remember those proteins, the little machines doing all the work? Well, their function is all about their complicated 3D shape. For 50 years, figuring out that shape just from the DNA sequence was one of the biggest, grandest challenges in all of biology. And then, an AI from DeepMind basically cracked it. This is a complete game changer for designing new drugs and understanding disease on a molecular level. This quote just nails it, doesn't it? It perfectly sums up the journey. We've gone from just reading a long, flat string of letters to being able to predict the complex, beautiful, three-dimensional machines those letters create. It's like going from a 2D blueprint to a working 3D model of the engine. And that brings us right here to today, and to a really big question for the future. With tools like CRISPR that let us edit the genome and AI helping us understand what those edits will do, we're not just passive readers of this code anymore. We are becoming the writers. So the question we really have to ask ourselves isn't just what can we do, but what should we do?